Why would an all-powerful, all-knowing God create genomes with more junk than functional DNA? And even more problematic, why would organisms that naturally group together possess identical or nearly identical junk DNA sequences at corresponding locations in their genomes? On the surface, the explanation that seems to make the most sense is an evolutionary one. Junk DNA sequences arose in the shared evolutionary ancestor and persisted in the genomes as the different evolutionary lineages diverged from a common ancestor. In other words, junk DNA sequences in our genome and the genomes of other organisms reflect our evolutionary history and can be used to map evolutionary relationships. Yet, over the course of the past decade, molecular biologists and geneticists have made discoveries that are forcing us to rethink this evolutionary view of the human genome. Bit by bit, researchers have discovered that most of the classes of junk DNA actually have function. And of course, if junk DNA is functional, it undermines the case for evolution. One could argue that these shared sequences of junk DNA that are actually functional, that occur in corresponding locations in genomes, actually reflect common design, not common descent. The case for design in the human genome virtually became stronger overnight thanks to the ENCODE Project. This project was initiated shortly after the human genome was sequenced. It became immediately apparent that simply having the DNA sequence for the human genome was not enough. There had to be some means to interpret the human genome. Nobody knew how to read the 3.2 billion genetic letters. We needed a Rosetta Stone for the human genome. Hence, the ENCODE Project was born. Its goal to identify all the functional elements in the human genome. ENCODE stands for the Encyclopedia of DNA Elements. This project began in the early 2000s. The pilot phase cost $55 million to carry out. The research consortium attempted to identify all the functional elements in 1% of the human genome. The success with phase 1 led to phase 2. Phase 2 cost $130 million and was completed in September of 2012. Phase 3 is currently underway, and the total cost for the ENCODE project will be about $300 million. This is actually a bargain because it cost $3.2 billion to sequence the human genome. So we'll be able to interpret the human genome for a mere tenth of the cost of actually sequencing the human genome. The ENCODE project was big science come to biology. The Phase 2 project consisted of 440 scientists and 32 research groups around the world who performed almost 1,700 experiments, analyzing 147 human cell types, generating 15 times 10 to the 12 bytes of data, and it required 300 years of supercomputer time to analyze the data. The ENCODE Consortium produced 40 publications distributed among some of the leading biology journals in the world. Phase 3 of the ENCODE project will survey the remaining 63 cell types and also look for functional DNA at different points in development and at different points in the cell cycle. This project will take us a long way towards understanding fundamentally human biology and human uniqueness. It'll help us to understand disease processes better. It'll help us to develop genetic tools that will allow us to diagnose genetic disorders. The insights coming from the ENCODE project also impact this creation-evolution controversy that we've been talking about. These results eliminate the best argument for evolution and eliminate the biggest challenge to biochemical design. So what did ENCODE discover that was so important? Well, these researchers performed 6 assays measuring transcription, the binding of transcription factors, histone binding, modified histone binding, methylation, and 3-dimensional interactions within the genome. All of these processes play a key role in what's called gene expression. It's one thing to know what genes are present in the genome. It's another thing to know how and when those genes are used. We can think of genes in the genome like being words in a dictionary. To write a novel, one needs to use the words in the dictionary in a variety of combinations, often using words more than one time. Each novel uses words from the dictionary in a different way to produce pieces of literature that communicate different meanings. The set of genes found in the human genome are like words in the dictionary. These genes can be used to build cells in the human body, with the cells functioning like novels. The genes are used or expressed differently from cell to cell, accounting for each cell type's unique features. Gene expression not only differs from cell to cell, it also changes throughout the course of the cell cycle and during growth and development. Each stage of the cell cycle, each stage of development represents a different novel that needs to be written. And here is the remarkable piece of the ENCODE project. It appears as if most of the DNA sequences found in the human genome are regulating gene expression needed to build and then maintain the human organism. Phase 2 of the ENCODE project reported that 80% of the human genome displays biochemical activity that likely reflects biochemical function. The ENCODE scientists expect that as Phase 3 is completed, 80% of the functionality will turn into 100%. Ed Young, who wrote in an article in Discover magazine, said this, and what's in the remaining 20%? Possibly not junk either, according to Ewan Burney. It's likely that 80% will go to 100%, and Burney serves as the head of the ENCODE Project Consortium. In other words, the human genome doesn't appear to be a wasteland of junk. It appears to be functional. Most of the DNA sequences in the human genome play a role in making us human beings. This insight stands as a radical revision of our view of the human genome. It's not a wasteland of junk, but an elegant biochemical system that is far more complex than we initially imagined. Well, how have biologists responded to the ENCODE results? Within hours of the publication of the ENCODE results, evolutionary biologists began to decry the ENCODE project, citing technical issues with the way the study was designed and the way the results were interpreted. These technical complaints continue today, igniting what are called the junk DNA wars between evolutionary biologists and genomic scientists. Evolutionary biologists argue that if the results of the ENCODE project are correct, then cornerstone ideas in the evolutionary theory, such as the C-value paradox, can't be correct. On the other hand, genomic scientists see value in the ENCODE results, using them to understand the genetic basis for disease. Evolutionary biologists have roundly criticized ENCODE scientists, claiming them to be incompetent and decrying the design of the ENCODE assays. Evolutionary biologists claim that if ENCODE is correct, then key aspects of the evolutionary paradigm are in deep trouble. But these critics are doing science backwards. Instead of data being used to evaluate a theory, the theory is being used to evaluate the data from the ENCODE project. The character of these objections are not lost on objective members of the scientific community, who have suggested that the real motivation for the criticisms of ENCODE are philosophical, even theological in nature. For example, two scientists, molecular biologists, John Maddock and Marcel Dinger, in an article published in the Hugo Journal, wrote these words. There may be also another factor motivating GRAR and other related articles, which is suggested by the sources and selection of quotations used at the beginning of the article, as well as the use of the phrase evolution-free gospel in its title. The argument of a largely non-functional genome is invoked by some evolutionary theorists in the debate against the proposition of intelligent design of life on Earth, particularly with respect to the origin of humanity. In essence, the argument posits that the presence of non-protein coding or so-called junk DNA that comprises greater than 90% of the human genome is evidence for the accumulation of evolutionary debris by blind Darwinian evolution, and argues against intelligent design, as an intelligent designer would presumably not fill the human genetic instruction set with meaningless information. This argument is threatened in the face of growing functional indices of non-coding regions of the genome, with the latter reciprocally used in support of the notion of intelligent design and the challenge to the conception that natural selection accounts for the existence and complexity of organisms. John Maddock and Marcel Dinger, who wrote these words, are not creationists. They're not intelligent design proponents. Our design of the human genome is in its infancy. Forced by their commitment to the evolutionary paradigm, Many biologists see genomes as cobbled together product of evolutionary history. But the more that we learn about the structure and the function of genomes, the more elegant and sophisticated they appear to be, and the more reasons to think that the genomes are the handiwork of our Creator. I would like to conclude with the words of Eric Green, the director of the National Human Genome Research Institute. During the early debates about the Human Genome Project, researchers had predicted that only a few percent of the human genome sequence encoded proteins, the workhorses of the cell, and the rest was junk. We now know that this conclusion was wrong. ENCODE has revealed that most of the human genome is involved in the complex molecular choreography required for converting genetic information into living cells and tissues. In light of the data coming from the ENCODE Project, as a Christian, I feel justified in viewing the human genome and hence human beings as a product of a Creator's handiwork.